Welcome all of you, dear students, in this session. Now we'll discuss about the membrane lipids. From the first session, we are talking about the chemical structure of lipid and biological function of lipids. Okay, so one of the most important structural function of lipid is that it acts as a major component of any biological membrane. Okay, so first of all, we have to know what is actually biological membrane, what are the main characteristic features of any biological membrane systems. So here you can see in a pointed manner, first of all, membranes are two layers, thick sheath, okay, like structure. So membranes are two layers thick sheath like structure formed by non-covalent assembly of lipid and protein. So it is obviously most of the membranes, like in cell membranes, are actually a two-layer thick structure, okay, which actually formed by lipids and proteins, okay, mainly consist of lipids, proteins, and carbohydrates are also there. But each of these particular biomolecules have different roles by themselves. Okay. Next, the thickness of membrane is near about 60 angstrom, or in nanometer scale, it is 6 nanometer to 100 angstrom or 10 nanometer. Okay. So membrane forms closed boundaries between different compartments. Okay. So first of all, what is the function of membrane? So membranes actually act like a boundary between different compartments. So next, it actually acts as barriers to the passage of polar molecules and ions. Okay, so it actually restricts the free passage of polar molecules, different kinds of varieties of polar molecules and ions to the inside of the organelles or inside of the cell. Okay, next, biomembrane are actually asymmetric. That means components of two faces are always differ from each other. That means as it is a two-layer thick sheet-like structure, but this two-layer is actually chemically asymmetric. These two layers are actually chemically asymmetric. That's why it is a very special features of biological membrane that biological membranes are asymmetric. That means components of two faces of two layers of the biomembranes are differ from each other chemically. And it has a huge significance, but it is not possible to discuss all the thing in this limited time. So very briefly, we'll see what are the different types of membrane lipids. So here we are focusing on the type of membrane lipids. First of all, membrane lipids have this particular structure, okay, hydrophilic and hydrophobic parts. So one particular membrane lipid, it has both hydrophilic or polar and a hydrophilic and hydrophobic segments. Okay, one end is hydrophilic and the other end is hydrophobic. Okay, so you can see this structure here. So if it is a schematic representation of one molecule of lipid. Okay, membrane forming lipid. Okay, so what is here? You can see this green head is actually polar head group, and this two leg-like structure, it is called as tail. Membrane lipid ka tail. Okay, non-polar tail group, non-polar trail group, and polar head group. Okay, so head group is like this, which is polar due to its chemical functionality, and there it has also two tail-like structure. Both of them, those are actually non-polar. So non-polar tail group and polar head group. So all the membrane lipids are actually composed of this type of membrane lipids are there, and it has two segments. One is polar head group, another is non-polar tail groups. Okay, so their hydrophobic interactions with each other and their hydrophilic interaction with water in cellular environment, okay, direct the packaging of lipids in the membrane. So if it is a single membrane lipid, okay, so multiple number of single membrane lipid assemble in a very structured, organized way, okay, although it is a totally a non-covalent association. Okay, so but they form this type of two-layer system. Okay, and it is actually part of it, it is actually a representation of our any biological membrane. Okay, so you can see all the polar head groups are arranged in a particular direction, and the hydrophobic tail groups are actually facing each other. That means hydrophilic head group are facing the outside cytosolic membrane environment, that means which is rich in water molecule, and it is actually interacting with the outside water molecules, and the inner part is actually formed by two layer of tail molecule, that means tail part. Okay, coming from this membrane lipids. Okay, so this inner part is actually highly hydrophobic, and the outer part in both ways are actually polar. Okay, so polar head groups are in this way, and the hydrophobic tail groups are in this way. Okay, so one end is hydrophilic and the other end is hydrophobic for each of one membrane lipids. Their hydrophobic interactions with each other, and their hydrophilic interaction with water direct the packaging of lipids in the membrane. Next, classification of membrane lipids. There are different types of versatility is there. So we have to classify. Membrane lipids are classified, can be classified based on their chemical properties of head group. So different chemical functionalities you can see in the head group. Depends on that, we can classify our membrane lipids. Membrane lipids are prokaryotes, eukaryotes, and archaebacteria are grouped into four major categories: phospholipids, glycolipids, sterols, and archaeobacterial ether lipids. So phospholipids, glycolipids, sterols, and archaeobacterial ether lipids. Phospholipid, glycolipid, sterols, and archaeobacterial ether lipids. So one by one we have to see. First of all, main class of membrane lipids. All are amphipathic. Okay, that three types of backbone in membrane lipids are you can find. That first of all, it can be made of glycerol. Glycerol, that means alcohol. Okay, which alcohol is present as backbone? So glycerol is made present. Okay, so it is a 3-carbon tri-alcohol. We all know trihydroxy alcohol. Okay, next the sphingosine may be there. Another one is the cholesterol. Okay, so here you can see this chart, membrane lipids. Okay, we can divide it into broader ways, that is a phospholipid and glycolipids plus cholesterol. Phospholipids may be of two types depending on the head group: glycerophospholipids, sphingolipids, and glycolipids also may be sphingolipids. Okay, so how it looks like? This is the structural representation. Here you can see in the glycerolipids, glycerol is actually forming the backbone. It is it is a tri-hydroxy alcohol, where the first hydroxy group is esterified with one fatty acid. Second hydroxy group is also esterified with another fatty acid. The third hydroxy group, you can see it is attached with one phosphate group as well as another alcohol. So this is the basic structural features of glycerophospholipid. Next, what is sphingolipids? Here you can see instead of glycerol as alcohol, you can see sphingosine is acting here as alcohol. Okay, so sphingosine ka one hydroxyl group is esterified with fatty acid, and another hydroxyl group is actually connected with phosphate and one choline atom. So one choline molecule. Okay, so here you can see again, in case of glycolipids, in case that means carbohydrate functionalized lipids, you can see sphingosine is again coming here as the alcoholic backbone, where you can see that fatty acid is attached with one hydroxy group, another one is attached with mono or oligosaccharides. Due to the presence of this particular carbohydrates, sugar, carbohydrates, or saccharides, it is called as glycolipid. Okay, so all sugar is known as saccharides, and saccharides sometimes it is represented as a prefix glyco. So all these are same. So due to the presence of this sugar molecule here, it is known as glycolipids. Next, we'll see what is phospholipid. Glyceryl 3-phosphate is a backbone here. Glyceryl 3-phosphate is a backbone here. In all phospholipids, here a polar head group is joined to the hydrophobic moiety by a phosphodiester linkage. Okay, the other two hydroxy group is actually esterified by fatty acids R1 and R2. These two hydroxy group will be esterified, carbon 1 and carbon 2 will be esterified with R1 and R2, and the C3 will be attached with phosphate as well as here phosphate and alcohol. Okay, so next we'll see the glycerophospholipids. Glycerophospholipids are also called as phosphoglycerides. Here it is the structure. You can see major lipid component of biological membrane consists of glycerol 3-phosphate, glycerol 3-phosphate. Okay, three carbon pair. You can see one phosphate group. Next, whose carbon 1 and carbon 2 is esterified by fatty acid. C3 is linked with another polar group. It is mentioned here as X. Okay, so glycerophospholipid is like this. In simplest glycerophospholipid, the X is hydrogen and it is called as phosphatidic acid. So what is phosphatidic acid? It is the simplest form of glycerophospholipid. What is glycerophospholipid? One type of membrane lipids. Okay, so phosphatidic acid is the simplest form when this particular X is hydrogen. Okay, now we will see how much variation is possible here. Only this X group is actually varying here. Okay, we can see here that when it is this X group is hydrogen, it is phosphatidic acid. When it is like this, it is called as ethanolamine. It is phosphatidyl ethanolamine. Phosphatidyl choline is there when X group is choline. Serine, phosphatidyl serine, glycerol, phosphatidyl glycerol. All these are different types of glycerophospholipids, and it is a very important structural components of biological membrane and lot of functions it has. Okay, so again the summary slide. Start with the glycerophospholipids. It is a very important type of membrane lipid. Start with the glycerol big backbone. It is a tricarbon alcohol. So one example is diacyl glycerol. It is a messenger molecule. C3 esterified must be with the phosphate group that gives the parent compound. Okay, the basic one, that is phosphatidic acid. Okay, where the X group is hydrogen plus another substituent also esterified to phosphate, ethanolamine, choline, serine, glycerol, inositol, phosphatidyl glycerol. All these we can find in that particular X position. Okay, so according to that, which particular extra group is there, we have to put the name in this way. Okay, phosphatidyl ethanolamine, phosphatidyl choline, phosphatidyl serine, phosphatidyl glycerol, etc. Next, we'll see the glycerophospholipids. Okay, it is actually forms when the esterifying results of esterifying different alcohols to the phosphate on C3. Okay, so here you can see that glycerol carbon 1, carbon 2, carbon 3. You can see carbon 1 and carbon 2 is as well attached with different two fatty acids, but carbon 3 here it is attached with the phosphate group. Phosphodiester kind of bonds are there, which is this particular phosphate group is holding in one hand the glycerol backbone, in another hand it is holding a particular serine molecule. Okay, one amino acid. That's why it is called as phosphatidyl serine. When this particular group is another ethanolamine, then it is called phosphatidyl ethanolamine. It is phosphatidyl choline. It is phosphatidyl inositol. Sphingolipids, when the backbone is another kind of alcohol, not glycerol. What is that one? Sphingosine. That's why it is called as sphingolipids. Sphingosine, the backbone of the lipid, that's why it is called sphingosine. Similar difference with glycerol-based lipids, now C1 has an OH group. Okay, so C1, carbon 1 has an OH group. You can see here C2 has OH group. C1 has an OH group. C2 has an amino group. C2 has an amino group. It is also 3-carbon compound. You can see Carbon 1, it has an hydroxy group. Carbon 2, it is contain one amino group. Another one is that instead of weight and glycerol fatty acid group in amide linkage, you can see this amine group is actually attached with, instead of weight, it is amine group. Okay, this particular nitrogen is conjugated with, amine group is conjugated with fatty acid, same like previous thing. So here you can see the amide linkage in carbon 2, where carbon 3 has weight group that does not get derivatized, and instead of one hydrogen atom on the glycerol C3 has a long hydrocarbon chain with one double bond. Okay, with one double bond, you can see it is a very long. Total carbon number is 13, 14, 15, total 15. Okay, 13, 14, 15 carbon long hydrocarbon chain, okay, with one double bond. Okay, so this is the structure of particular sphingosine. This is the general structure of sphingosine. Ceramides is an example which have an fatty acid in amide linkage to the amino group of carbon number 2. This is in all sphingolipid ceramides. Okay, next we'll see the cholesterol, the last component of membrane lipids. What is cholesterol? What is the structure of cholesterol? It is actually 4 fused hydrocarbon rings are there. 1, 2, 3, and 4. Okay. The third one with 6 carbons. First one with 5 carbons. Okay, is planar, rigid, electrically neutral. It is amphipathic totally, and mainly in plasma membrane of animal cells. Organelle membranes generally have less. It majorly found in the plasma membrane of animal cells. Organelle membrane generally have less amount of cholesterol, and it is rarely found in bacteria. Okay, so what are the typical functions of cholesterol molecule? Important membrane constituent, influences the fluidity of biological membrane. So you have to remember this word, fluidity of membrane. So cholesterol actually influence the fluidity of biological membrane. Next, precursor of bile acids. It is a precursor that means each and every biomolecules, there is a biosynthesis procedure. Bile acids also has a biosynthesis procedure, and this particular cholesterol is the precursor. That means a parent compound from which this is formed through different chemical reaction, biochemical reactions. And action of and the function of bile acid, it is the famous emulsifier, which can emulsify the large granules of fats, okay, into smaller volume emulsions. Okay, next, the precursors of hormones, that is steroid hormones. Okay, so these are the particular functions of cholesterol. First one, in important membrane constituent. It is the precursor for the bile acids biosynthesis. It is also a precursor of steroid hormones. Okay, thank you, students.